This is the Luz and Guattari, and they are two agouti Mongolian gerbils. This video will explore the lives of these two gerbils in order to elucidate the ins and outs of the vertebrate, nervous, and musculoskeletal systems. Batchelder et al. Described the Mongolian gerbil as a docile rodent species that is native to northern Africa, India, Mongolia, southwestern and central Asia, northeastern China, and parts of eastern Europe. It belongs to the class Mammalia and order Rodentia. The breed that is popularly available as a pet in the United States is the common agouti, or mixed brown gerbil, which is the breed that we see in front of us. These gerbils are descended from 20 pairs of the animal which were originally captured for domestication in 1935. As a result, gerbils available commercially typically have a very low degree of genetic variability. Mature gerbils are typically smaller than rats but larger than mice, with males tending to be larger than females. Both the gerbils in our video are males. They have broad short heads with prominent ears and large eyes typically found in nocturnal animals, and their vision is very well developed. Their excellent sense of smell allows them to use a variety of odors as discriminative social cues. Gerbils are well adapted for dry environments, with high heat tolerance and the ability to thermoregulate well. Additionally, they require very little water, obtaining enough from their diets. This results in highly concentrated urine, due to the nephron loops in their kidneys having a high ratio of long loop to short loop nephrons. Behaviorally, gerbils are docile animals that have a curious and exploratory nature, as demonstrated by the propensity toward increased activity when placed in an open field. They are social as well, typically living in burrows that they create through their compulsive burrowing behaviors. They tend to be most active at night, but are often awake during daylight hours, exhibiting bursts of activity between periods of rest. Gerbils are highly social creatures, and they exhibit territoriality by marking with secretions in their ventral sebaceous gland. This marking can be used for demonstration of social dominance, exploration, and demarcation of territory. Additionally, when gerbils are mating or excited, they thump their legs rapidly to signal to others. Now that we're a little bit more familiar with our furry friends, let's dive into the mammalian nervous system, with these two as our guide. To develop an understanding of the regulatory and sensory functions of the mammalian nervous system, we must understand its functional hierarchy, the role of the neuron, the conduction of signals, and the function of neurotransmitters. In mammals, the nervous system is broadly divided into two parts: the central nervous system, which consists of the brain and spinal cord, and the peripheral nervous system, which consists of the peripheral nerves that extend from the spinal cord. The peripheral nervous system is further subdivided into the afferent or sensory division and the efferent or motor divisions. The afferent division is responsible for conducting sensory information towards the brain and spinal cord, whereas the efferent division is responsible for conducting information away from the spinal cord towards the peripheral limbs and the viscera. Furthermore, the efferent or motor division is divided into the somatic and the autonomic nervous system. The somatic nervous system permits conscious control of skeletal muscle, whereas the autonomic nervous system regulates involuntary muscle actions, like those needed in cardiac muscle or in the smooth muscle of the intestine. The autonomic nervous system is further divided between the sympathetic and the parasympathetic nervous systems. The sympathetic nervous system broadly prepares the body for stressful situations or behaviors that will require energy expenditure, whereas the parasympathetic nervous system relaxes the viscera. These systems and subsystems represent a hierarchical organization that arises out of vast systems of interconnected neurons. The distinction between the sensory and motor divisions of the peripheral nervous system can be shown by the gerbil's quick response to an outside stimulus such as a noise, or in this case, the presence of my hand, to which the gerbils look up and acknowledge me. The sensory division of their peripheral nervous system processes my hand and my presence in their sight, whereas the motor division encourages them to look up it and acknowledge me. In order to further understand the nervous system, it is crucial to understand the function of neurons. Parker et al. Broadly defined neurons as electrically excitable cells that transmit signals throughout the body, which utilize electrical and chemical components in the transmission of information. The nuclei/ganglia of these cells are arranged in functional circuits within the nervous system that contribute to specific behavior or information. The connection between these neurons are known as synapses, wherein the membrane which conducts the signal, the presynaptic membrane, is positioned near the receiving membrane, the postsynaptic membrane. Typically, the signal-conducting membrane is an axon terminal, whereas the receiving membrane can be an extension of the neuron nucleus known as a dendrite or a muscle. In this case, we have what's known as a dendritic spine receiving the signal. The electrical conduction that occurs through neurons is known as an action potential, where the membrane potential of a neuron is temporarily reversed from its resting potential of negative 65 millivolts up to positive 40 millivolts. An action potential will only occur if the membrane depolarization reaches a voltage threshold, meaning that the transmission of electrical signals throughout the nervous system follows an all-or-nothing schematic. The electrical signals involved in the transmission of information throughout the central and peripheral nervous systems can be transmitted through two major methods: direct electrical coupling and chemicals known as neurotransmitters. In the first instance, neurons are directly coupled to one another through cells known as connexons, which form gap junctions. These cells are unique to chordates, allow for bidirectional flow of ions, can rapidly change cell membrane potential, and only produce excitatory signals. Here we can see the connexons, two bound to one another which form a gap junction. On each membrane, one connexon has 6 subunits, which connects to the connexon on another membrane. By contrast, neurotransmitters produce a signal by synaptic vesicle release to receptors on the postsynaptic membrane. These neurotransmitters flow in only one direction, can produce both inhibitory and excitatory responses, and their intensity can be modulated over time. Neurotransmitters bind either to ion channels or G-protein coupled receptors. Here we see our ion channels, and this represents a G-protein coupled receptor. Ion channels produce effects in the postsynaptic membrane by changing membrane permeability to ions, either increasing or decreasing it, while G-protein coupled receptors produce effects via second messengers. This G-protein decouples and then induces many effects. In vertebrates, most synaptic communication involves neurotransmitters rather than electrical transmission, and the primary excitatory neurotransmitter is glutamate, while the primary inhibitory neurotransmitter is GABA. Gerbils, in a way that is common to all vertebrates, receive signals from their environments and react. Neural signaling is crucial to their ability to process sensory information and perform the appropriate behavioral response. Behaviors specific to gerbils include boxing, foot thumping, scent gland marking, and burrowing. Thus far, we have discussed the basic principles underlying the interactions of neurons in mammalian nervous systems, but we will now move to the ways that the nervous system interacts with muscles to produce mechanical behaviors. The types of movements available to vertebrates fall under the categories of voluntary, reflex, and rhythmic motor pattern movements. Voluntary movements are those which result from conscious control, whereas reflexes are involuntary and unconscious. Rhythmic motor patterns act as a kind of middle ground between conscious control and reflex movement in the sense that they involve conscious initiation and termination, but do not require conscious control once the movement has started. In gerbils, motor patterns can be seen in burrowing, running, and foot-thumping behaviors. As you see Deleuze run up my arm, the running motion that he enacts is an example of a rhythmic motor pattern. Broadly, movements in vertebrates come about because of neuronal control of skeletal muscle contractions. The initiation of these contractions comes from motor neurons with origins in the central nervous system and terminations in the peripheral nervous system. These neurons fall into the category of either upper or lower motor neurons. In the gerbils, the upper motor neurons are found in their brain, and they do not directly innervate the peripheral nervous system. Merely they carry commands from the brain to the lower motor neurons. Here, as they fight with one another, their upper motor neurons are telling their legs, the neurons that innervate their legs, to move and to chase one another. Lower motor neurons, by contrast, have cell bodies in the brainstem and spinal cord and directly innervate the periphery of vertebrate bodies. All movement in vertebrates comes from lower motor neurons. One of the most important principles in vertebrate movement is the concept of the neuromuscular junction and excitation-contraction coupling. While I will not go into excruciating detail so as to get the overall picture, I will explain how these two are interconnected. Starting at the neuromuscular junction. Calcium ions bind to receptors on a neuron which innervates the muscle fibers. This calcium moves to synaptic vesicles within the neuron, and they bind to what is known as a snare complex. Much research still needs to be done on the snare complex. However, it is known that it helps with synaptic vesicle movement towards the cell so that neurotransmitters can be released by exocytosis. In this case, the neurotransmitter released is known as acetylcholine, and it binds to receptors known as nicotinic acetylcholine receptors. These are ionotropic receptors, and they allow for the influx of calcium into the muscle. Once the calcium is in the muscle, it interacts with protein fibers known as myosin. This entire complex is known as a sarcomere. It contains the Z disc, to which the actin fibers are attached, the A band, which has both actin and myosin fibers, the H band, which has just myosin fibers, and the I band, which has just actin fibers. The guiding principle of these filaments is what is known as the sliding filament theory, wherein the myosin chains attach to the actin and pull it closer, causing the Z discs to move toward one another. This is the principal way in which all vertebrates contract their muscles, and it is triggered by calcium influx into the muscle through acetylcholine. Today, we began by discussing some of the unique physical and behavioral features of Mongolian gerbils. Next, we explored the major subdivisions of vertebrate nervous systems as a whole. Additionally, we explained the major physiology and function of neurons, including action potentials, electrical versus chemical signals, and excitatory versus inhibitory signals. Finally, we briefly discussed the different types of movements in vertebrates, neuronal control of skeletal muscle, the neuromuscular junction, and the sliding filament theory. We have covered a lot of ground today in regard to both vertebrates and gerbils. In concluding, I hope that I have left you with a greater appreciation for the complexity and interconnectedness of both gerbils and vertebrates in general.